Hello. Hello. Hello.
the button. Fantastic.
I was listening.
So welcome everybody, and it is my great pleasure and delight to welcome Craig Jenne to UBC and to the CBR. Known Craig for quite some time. He's got a great background. So he trained with some of the best immunologists in the world, beginning with John Reynolds, who's an ex-grad of the Basel Institute for Immunology and a fantastic mentor. That's where Craig did his PhD. And then he went on to a pretty amazing postdoc that was co-sponsored by Chris Goodenow at the Australian National University in Canberra, looking at E and mu mutagenesis as a way of generating mutations that influence immunology and immune response, and co-working with Jason Sister at the University of California, San Francisco, who's a founding father of kind of stromal cells that influence immune development. From there, Craig came back to Calgary and did a second postdoc with the great Paul Koobs, who you might know for intravital microscopy work, and really started that journey on looking at platelets in immunology and immune response intravitally. And that's a really selective niche and one that's been pretty much underexplored, but I think a great place to work. And I think a lot of the folks in the room would be really interested in hearing what he's going to tell you next. Craig's also had a lot of leadership roles, so he's a full professor, also the deputy director of the Snyder Institute for Chronic Disease. He's played a major role in the Canadian Society for Immunology on both the EDI committee and now policy committee that's trying to get government more interested in how science can guide good governance. So with that brief introduction, I'll turn it over to Craig. Please welcome him.
Thank you, Kelly. It's really exciting to be here today. I can't believe this is my first visit ever to UBC, and I'm just completely blown away by campus and the people I've chatted with already. So it's wonderful for the invitation, and I'm so happy to be here. What I'm going to present today is a few perhaps disconnected stories on the surface, but it's more of a journey on how we've started to tackle some questions about platelets and immunity, and we're going to show some published data that establish the questions we're asking now, some fresh off the press data that we're still, hopefully within the next few months ready to submit, as well as some failed data. And for the trainees, not everything always works the way we plan it. And ultimately, I think where we're coming out of this at the end is our initial understanding, or what we set out to understand, may not actually be at all what's happening. And we've been misreading a lot of the tea leaves now for a little bit. So I want to try and discuss how we've maybe changed our direction a bit. So my lab is an immunology lab. We study the host response to infections. And we set out early on, this was during my postdoc with Paul Koobs, to really ask, what happens when you actually become infected? And we know that you'll eventually clear the infection, but what's going on in the minutes to hours? And if we think about the immune response, we know that there's an immediate, rapid, innate immune response. And then as that wanes, we see the pickup of the slack by the adaptive immune response in the days following. And we wanted to focus early on, and in fact, focused right at the very beginning of this process. And we developed a really simple model. I mean, it's almost hard to call it a model, but we simply inject our mice with Staph aureus intravenous, and we look at what happens to the actual bacteria. And as a classically trained immunologist, I mean, the answer is obvious. Your spleen is your filter for your blood, and it's full of macrophages. And so if we track where the bacteria go, we were quite shocked to see essentially nothing goes to your spleen. And this again is challenging for me as somebody who believes the spleen was the center of the universe. Where the bacteria actually go are to your liver, and this again struck me at the time as not right, and really needed to dive into this. So we started applying our favorite technique, intravital microscopy, to this and ask what happens to the bacteria. So if we look in a mouse liver, and we'll see if the laser's working, all the red cells here, these are Kupffer cells. So these are liver resident macrophages. They live in the blood vessel. So they're not in the parenchyma, they're actually in the vessel lumen. And if we were to inject a fluorescent Staph aureus, So this is about a 7-minute video time lapsed, and we see that the liver immediately filters the blood through these Kupffer cells of the Staph aureus. And this is happening in seconds. This is not an adaptive response. We don't need germinal centers. We don't need B and T cells. Right. So what about the other cells, right? What are the neutrophils doing? So in a healthy liver, we get a few neutrophils. These little blue dots are neutrophils in this living mouse liver. If we look 4 to 8 hours after infection, we get a lot of neutrophils. So this is the classic innate immune response, right? You get an infection, the first few hours, these cells are activated, they recruit. If it's skin, you get a pussy type infection. The liver, they're crawling around and hopefully killing bacteria. But when we sat back down and looked at our simple little model, it created a bit of a problem that we had to address. And that is, if we imagine infection happens at time 0, and this peak neutrophil response is happening at 4 to 8 hours, there's a substantial window in here that microbes can exploit, in particular bacteria. I mean, they replicate every 20 minutes. If we're talking about waiting 8 hours for neutrophils to come to the rescue, you know, there's potential problems. So we could imagine, for example, our Kupffer cell is going to catch some bugs, hopefully eat them. But you know, if there's a lot of bugs, they start accumulating and replicating faster than the Kupffer cell can actually process them. So there has to be some other mechanism. Otherwise, the predicted outcome would probably not be good for the mouse. So we started asking ourselves, well, if we need an immediate response, are there cell types that we might not have to recruit? Are there things that are always present, always in the blood, and at substantial enough numbers that we don't have to bring them in from the bone marrow? So we were looking at platelets for other reasons. And what we started noticing was that if we were to inject bacteria, so in this case, the red are platelets, bacteria are still green, but as the liver catches bacteria, so for example, we catch one here, the platelets start accumulating on the surface and around those bacteria. So we don't have to wait for neutrophils. These are found in our blood at a billion per mil, and they're immediately present. So this got us wondering, is this part of the host response as opposed to collateral damage? And we started asking the question, well, maybe platelets are actually primary immune cells. And if we go back through literature and early immunology work, you know, we actually recognize platelets or their precursor, hemocytes, in invertebrate as the primordial immune cell. So in invertebrates such as sea urchins or starfish, neutrophils and platelets are the same cell. So they're phagocytic platelets that when they eat something, they can actually clot, but they can oxidative burst. They can have lysosomes and digest bacteria. So we're asking, well, maybe platelets have retained this ancient role. And if we're looking once again at this bacterial capture model, we could see within a matter of 3 minutes, 4 minutes, we get this massive platelet dome sealing off the bacteria, very reminiscent of how a hemocyte encases a bacterium in a starfish or a sea urchin. If we disrupt this, if we prevent this aggregation, so we use a GP1B alpha deficient mouse, we start to see massive tissue damage. So this is a dye that is cell impermeable. So what we're seeing is propydium iodine labeling of the endothelium. So in the absence of these platelet domes, we're seeing endothelial cell death. And functionally, if we measure that, we can see that there's liver damage occurring. So ALT is going up in the mice, again, that cannot form these aggregates, the GP1B deficient animals. And ultimately, if we get rid of platelets, if we deplete them and we do the same infection, the mice simply don't survive. So that this immediate platelet response is absolutely critical. And we've gone on to show that this is largely about sealing off these bacteria. If we allow individual platelets to dock, it's not sufficient. We need the full domes to encase the bacteria in order to essentially buy time for neutrophils to come and clean up the mess. So this early finding got my group really wondering how are inflammation and coagulation linked. So these little platelet domes aren't actually clots, but they have all the hallmarks and all the players that might be needed. And this really raises the questions about things such as immunothrombosis, and more recently our labs have been thinking about thromboinflammation. So if we look at human disease, septic patients in particular, we tend to find dysregulation of the coagulation system. And in fact, if you develop disseminated intravascular coagulopathy or DIC, this is strongly associated with bad outcomes. So we know that infectious disease and inflammation are tied to clotting, and that these clots can cause problems. At the same time, the coagulation cascade feeds back onto the immune system and activates more immune cells. And this is what we term thromboinflammation. So we're seeing this linkage, and we're trying to dissect it. And I will admit, coming into this, we were very excited about the concept of immunothrombosis. So is it the immune system that is causing this clotting? So if we look a little later in the immune response, so this is now, again, a control animal to show what platelets are doing under basal conditions. If we look 8 hours instead of 8 minutes after the infection, and it doesn't matter if this is bacterial or viral, the phenotype is very similar, we get massive platelet recruitment to the liver. So we see these huge aggregates. And what struck us is that initially we thought this might be clotting, but if we look closer, we find out it's actually not true thrombi. These platelets are not forming permanent structures. They're individual platelets that are associating with neutrophils, and we can almost imagine they're rolling on the surface of the neutrophil. If we do a 3D model, I think it's even more clear that we can see neutrophils binding on the upstream side, moving along the membrane, forming an aggregate on the downstream side, and then re-releasing back into circulation. So these are not clots per se, but they're very strong neutrophil platelet interactions. So if we, in this case, this is a poly-IC treated animal, so a viral mimic, we get these massive aggregates on the platelets. So if they're not clots, then what are they actually doing, right? These aggregates are obviously being actively formed. And at the same time, while I was working on this, other people in Paul's lab were following a different neutrophil response, and that was neutrophil extracellular traps. So were these platelets contributing to traps? So what are these traps? So as a postdoc, I was given the wonderful job in Paul's lab of wearing a pager, which meant when it beeped, I had to go. So for those who wonder what Paul is like as a supervisor, I wore a pager, and when Paul beeped, I went. So like one Sunday morning, I get paged to go over to Foothills Hospital, and we were draining abscesses from an elderly gentleman. So Sunday morning, I sit there with my 50 mil Falcon tube and collect what's coming out of the poor guy's abscess. So fun times. And then you get run back to the lab, and you spread them around on slides, and you stain them, and you can get pictures like this. So if we stain for neutrophils, we can see the abscess is full of these red cells. If we do a pan-leukocyte marker, right, we can see, again, almost everything is a neutrophil. But when we stain for DNA, we get these two distinct patterns. We get this wonderful trilobe nucleus stain and this weird cloud. And when we overlay them, we can see our prototypic neutrophils, nice trilobe green nucleus in the red labeled neutrophil. And then we can find these cells that have 3 black holes where they used to have a nucleus. And then surrounding the cell is this wonderful net or web of DNA. So this is really, we've always learned that pussy abscesses are neutrophils. They're also largely extracellular DNA from these cells. What do they do? Well, looking in the mouse liver again, we can find neutrophils that are releasing these DNA structures. So the red is DNA, and we can actually see it lining and outlining the blood vessel wall. We can see the tube coated with DNA. And in this DNA web are caught bacteria. So these are anti-pathogen mechanisms. They ensnare bacteria. At the same time, this is when my lab was starting its own venture. And in the literature, there was a lot of reports about components of nets, histones or individual things. If you put them in a test tube with blood, the blood will clot. But we were wondering, does this happen in vivo? And this required a slightly different approach. So we had to develop a way to actually see blood clotting. And what we sought to do was not to look for the presence of the enzymes, but to look for the enzymatic activity. So thrombin, for example, exists in your blood as prothrombin. When it becomes activated, it starts cutting things such as fibrinogen to make fibrin-based clots. So we were able to adapt a probe that has a fluorophore in it, this little gray square, and on the end of the peptide is a fluorescent quencher. So it's absorbing all the light. And linking the two is a very specific enzymatic cut sequence. So a peptide that is recognized and cleaved by a given protease, in this case, thrombin. And in the presence of active thrombin, we'll clip that linker, we'll release the quencher and reveal fluorescence that's present in the molecule. So with this, we sought to see, do we see blood clotting in sepsis? And we used our mouse model of bacterial infection in the liver. So lots of neutrophils recruited. And when I start the video, we're going to be injecting this probe in real time. So we'll see this essentially unfold in real time. And here is the amount of thrombin activity in your liver in sepsis. So almost every sinusoid is showing active thrombin. We can quantify these, we can take still images, and again, it's quite striking and remarkable how much thrombin activity is in the liver. What was fascinating to us is most of this is actually closely associated with staining for nets. So we are seeing the nets perhaps driving it, and we know if we get rid of the nets, the thrombin signal disappears. But thrombin itself isn't a clot. So we also sought to try and determine what is this protease doing? And we can look for deposition of fibrin, and we can see in very much the same pattern, microvascular occlusion with fibrin-bearing thrombi. So we do see a linkage now between this early response where platelets are driving neutrophils to do something. The end product, the neutrophils release are nets, which in turn generate thrombin and now feeds back into the system. Ultimately, what do these little fibrin structures do? Well, if we inject a contrast agent, we can see that the vessels containing thrombin are not perfused by our contrast agent. So they are creating ischemic patches in our organs, and we can go ahead and quantify this. So again, if we add DNAs to the system, get rid of the nets, our thrombin signal goes down. If we use a PAD4 deficient mouse that can't release a net, we don't get thrombin. And ultimately, if we use these animals that can't release a net, we don't get liver damage. We've gone on and confirmed this with anticoagulants as well. So if we administer our gatroban, we again fully protect the liver, even though the nets are still there. So we were able to start to put together that part of immunothrombosis is likely due to these intravascular nets. One of the questions that we were asked, it was actually very early on, is, well, we have a lot of drugs that target platelets, and what happens to these people? So the argument is that immunothrombosis is part of your innate immune system. It limits bacteria from disseminating. So we need these little clots to stop bugs from moving around. And by that extension is that if somebody's on an antiplatelet drug, well, they must have more infections. So we started trying to test this. So we know that the nets can get the pathogens. We know that the nets generate thrombin. And in theory, those thrombi restrict pathogen dissemination. So we set up a model, again, looking at our favorite organ, the liver. So here's our healthy animal. Here's our animal with Staph aureus sepsis. Lots and lots of platelets, lots of nets in the cyan. So exactly the same thing we'd seen before. But if we treat our animals with aspirin, either low dose prophylactic or even a therapeutic dose after infection, what we see is quite striking. So platelets no longer recruit to the liver, and critically, we don't get nets. So if the platelets aren't recruiting, they're not talking to the neutrophils, we're not getting nets. So this must now absolutely lead to bacterial dissemination. So we start testing our model, and we find that not only do we not get nets, we get less non-perfused areas in the liver, and we get a reduced ALT, so liver damage. So this is highly protective. But when we look for bacterial counts, and we've done a bunch of organs, I'm only showing liver and lung, we find no difference whether you're pre-treated with aspirin or you take it therapeutically after infection in the liver, in the lung, in the spleen, in the blood, that we don't actually impair pathogen clearance. So these immunothrombi are entirely dispensable, at least in a healthy individual that has other working immune systems. So as a summary of this part, although we still believe immunothrombi probably have a time and place, for the most part, they're dispensable and they're causing more damage than harm, at least in pathogen control. If we add to that, in most clinical settings, we can get antimicrobials on board very quickly, that we really don't have to worry about bacterial dissemination if you're presenting at a hospital environment. So really, we need to be working at what are these immunothrombi, how do we stop them, how do we break them down? And we'll come back to this in a little bit. We sought to see if this was entirely a liver-specific thing. So we started moving into other organs, and the next logical one for us was the lung. And again, we can apply intravital microscopy to the lung. I've done some work, and I'll show a little bit of data on bacterial infections of the lung. But at this point, we also chose to change to a really pathogen close to my personal interest, which was viral. And we moved into a mouse model of influenza. So we wanted to understand what's happening. And this work started before the pandemic. To put things in perspective, influenza was the leading cause of death in Canada from infectious disease prior to the pandemic. So this was a clinically relevant disease to understand. Again, if we apply our intravital microscopy and we give these mice flu and we look on day 5 after flu, we started tracking all the usual players. We get neutrophil recruitment, we get platelet recruitment. Critically, we get net deposition in the lungs. So the little red is extracellular histone. So we are seeing the classic model we saw in the mouse liver all play out again in the lung. The hypothesis would be if we're getting strong nets, we should be seeing thrombin. And again, if we inject our green probe, we get diffuse thrombin throughout the lung, and we can measure that. If we give a thrombin inhibitor, we quench the probe, or we fail to generate the fluorescent probe. So everything is matching the initial paradigm we set up in the liver. Again, we wanted to understand, is this fibrotic clots or not? So we stain for fibrinogen and fibrin. And we can once again see these wonderful little structures in the vasculature of the lung that we can't, unfortunately, the current antibodies we have available cannot differentiate between fibrinogen or fibrin. But we can see in a healthy animal, they're not accumulating. In the infected animal, we get this robust fibrinogen deposition. And again, ultimately, we wanted to look at something functional. So our intravital images give us a really good idea of what's happening in this microenvironment. So in our control animals, we can see individual green platelets. The lung is quite impressive in that it has a resident neutrophil population or marginated neutrophils that are there all the time. But these guys are pretty benign. They're not clumping. They still move around a bit if we give them flu, and we get these massive occlusions, all these little dark spots or shadows of other leukocytes that are not labeled and can't move through the blood. So we can see how devastating influenza is. So again, massive platelet aggregates and occluded vessels. So at this point, we were pretty happy. Everything was matching exactly what we had previously mapped out. And if we were to put together a lung model, we'd be looking at something similar to what we had in the liver, where neutrophils get recruited. Platelets will then dock on this neutrophil. We'll get a net release. That net will generate thrombin. And then from there, we can get clots or immunothrombi. So we're pretty happy, pretty comfortable with this. And we started to dive a little deeper, which was the worst thing we could have done. We should have just said lungs are like liver, published the paper and left. But instead we decided to run some, at the time we thought they were experiments for fun. Turns out they weren't. So we asked, what's more important, this initiation or the feedback loop? Because what we're generating is a lot of thrombin, and we've been focused on the immunothrombi. So we've been focused on this end product. But we had sort of ignored the fact that thrombin feeds back on this system, right? So thrombin activates platelets. And in fact, the principal receptor on a platelet was originally known as a thrombin receptor. So these are the protease activated receptors. So we started asking, well, can we maybe get in here and break this? Is this how we cure flu patients? If you're sick and end up at the hospital, can we tone this down? And we'll still get our protective response, but we're not going to get amplification. So we took two approaches. One is we could block the thrombin itself, again, with our gachuban, so a thrombin inhibitor, or we have a PAR4 receptor, which is predominantly on platelets, not exclusively, but it is the dominant thrombin receptor on mouse platelets. It's a little different than humans. Humans use PAR1, mice predominantly PAR4 on their platelets. So we were fortunate that we were working with Dr. Morley Hollenberg at the time in Calgary, and he is Mr. PAR and had every antagonist known to man. So it was a quick trip down the hall, and we had tubes of PAR4 antagonists that we could quickly try. So when we did this, we're pretty happy with the initial results. So if we block, in this case, PAR4, the same was true for the argatroban, we saw a significant drop in what we measure as a wet-dry ratio of the lungs. So we harvest the lungs, we weigh them, we put them in the oven over the weekend, we weigh them again, and we can calculate what percentage of the lung was liquid. So when animals are quite sick, they get edema and fluid in the lungs, so their wet-dry ratio goes up. And when the animal's less sick, that wet-dry ratio comes back down. Blocking PAR4 returned the lung fluids entirely to baseline. So it was highly protective. If we looked at that feedback loop, we wanted to slow the platelets down. Again, really encouraging results. So whether we block the receptor on the platelet or whether we block the thrombin, we can prevent platelet aggregation. So across the bottom are the size of platelet aggregates. So in an infected animal, we're seeing 100 square micron aggregates of platelets. These are completely inhibited if we block thrombin or PAR4. So again, very happy with this. Until we counted our neutrophils. And it turns out if we interrupt this signaling pathway, we don't recruit neutrophils to the lung. So we're not stopping amplification. We're actually preventing the earliest thing from ever happening. So if neutrophils are making a net and the net is generating thrombin and thrombin's feeding back, how did we stop neutrophils from arriving before they made thrombin? Was the problem the lab was dealing with. And I mean, there was no way to get around it. It was essentially that thrombin generation comes before neutrophils get to the lung. And we know that because the argatroban we administer has a half-life of about 12 hours. So neutrophils show up on day 2. The drug is already out of the mouse's system. There is a thrombin event that's happening in the first 12 hours that actually sets up neutrophils to arrive. So we go back to our little model here. It meant we're missing something, and there has to be a thrombin signal somewhere else upstream. And we know that it's acting through PAR4 because PAR4 was equally protective, or inhibition of PAR4. So we tried to figure out where could this possible signal come from. And we looked at, again, typically the usual suspects on this. And the most obvious one was perhaps we were expressing tissue factor. So this is the stimulator of one of the 2 coagulation pathways. So this is a protein that is expressed on damaged endothelium or sites of damage and can generate active thrombin. So we went back into our mouse model, and here's our control with individual green platelets and a few neutrophils. If we give flu, we get these large platelet aggregations. And if we give a tissue factor blocking antibody a couple hours before we administer flu, we can completely inhibit platelet aggregation. If we look at the overall outcome of disease, so again, administering tissue factor blocking antibody, the animals lose less weight here in green. The animals have fewer neutrophils per field of view by intravital imaging, and critically, the animals do not produce nets. So again, we've not touched the neutrophil itself. We have simply blocked the ability to generate thrombin. So this was quite exciting for us, that there's an early tissue factor signal that must be happening that sets up this 5-day influenza. And in lab meeting, it became very clear to us, I mean, should have been clear to us, but from recent comments from the Alberta government, it's apparently not clear to a lot of people that viruses are not bacteria. They're apparently different. So we thought, well, this must be a unique thing to viruses, right? Viruses are infecting inside our cells and triggering tissue factor expression. And our model in the liver with Staph aureus doesn't use tissue factor. So we then tried Staph aureus in the lungs, and again, blocking tissue factor. And we're quite shocked to find in the lungs for bacteria, tissue factor was also highly protective, or tissue factor blockade. Again, animals lose less weight over the first day of infection. We see fewer neutrophils, and we see almost no nets produced in an animal that is receiving Staph aureus, but has tissue factor blocked. And now we started to panic, right? This is, how did we miss this in the liver? How bad were we? So we went back to the liver saying, well, is thrombin required to initiate everything we see in the liver? So we administer our tissue factor and give an intravenous Staph aureus infection and then track the response in the liver. And it turns out tissue factor is entirely dispensable in the liver. So this is a lung-specific event that doesn't matter if it's viral or bacterial. And we've now tried 3 different bacteria. They always generate a tissue factor response. That response initiates some form of thrombin-based signaling, and we drive inflammation from that point forward. In the liver, it's not. And this might be due to the fact that the liver does have things like Kupffer cells that are remarkable sentinels without having to engage a thrombin pathway. So once again, coming back to our little model here, now we understand that upstream, there's a tissue factor, there is a thrombin spark, which then we still believe acts largely on platelets, but might not. I'll come back to that in a second. And then we can kick off our classic model. But as I said, there may be other cells in here. So just because we blocked PAR4, which is predominantly on mouse platelets, it's not the only place. So studies by the Mackman group at UNC had also shown that the epithelium has PAR4, and that is required to upregulate things such as TLR3 based signaling. So we're currently trying to dive into what is the effect on platelet versus other cells. And if we think down the line, again, thrombin is not restricted only to PAR4. We know PAR4 is important because we blocked PAR4, but thrombin could have effects through the epithelium, endothelium, monocytes, neutrophils, anybody with a PAR receptor in particular. So at the end of the day, this does set up a bit of a problem for us. And that is, if we focus entirely on the neutrophil, we've got a couple of key players, thrombin and platelets, that are initiators, but they're also effectors, and they're also amplifiers. So it makes it very difficult now to intervene. And how are we therapeutically going to target this thing that we can break the cycle or at least restrict the inflammation so that we can control the disease? And it's also this fact that everything is driven upstream and not dependent on the actual thrombus formation per se, that we've been chasing this for about 5 years now as immunothrombosis. But I do think that we're probably more likely studying thromboinflammation that has thrombosis as a potential output. But the real problem, or at least the real therapeutic opportunity, is targeting the other thrombin-mediated signals, not the fibrinogen deposition. So we needed at this point to take a step backwards to try and figure out how we're going to address multiple players doing multiple roles and find a usable therapeutic target. Our argatroban was exciting, but it had to be administered at the time of infection. If you wait even 1 day, it has no effect. So there's an early signal. As far as the therapy, that's not going to work. People don't go to the doctor saying, I'm going to get flu tomorrow. Can I get argatroban today? So we needed to find something downstream. So we went back to this question here. And that was when we're watching this video, we can clearly see we have our neutrophils in red, we have our platelets in green, but we have no idea what they're doing. I mean, the neutrophil's sitting here, but is it doing anything? Is it producing cytokines? Is it talking to another cell? We have no idea. Likewise, we have no idea what all these little shadows are. Are these all red blood cells? Are there? So we had to approach this differently, and intravital wasn't going to take us to that next level. So working with Tony Dufour in Calgary, we took a step back and tried to rethink this maybe through a proteomics approach. What's happening in the lung? And to get this started, we actually, really simple, we're going to harvest tissue, we're going to do mass spec, we'll generate proteomes. To get started, we want to work with a single cell. We want to work with lung epithelium from a mouse. I'm very bad at cell culture. If you ever want to see wonderful fungal colonies, come check out my cell culture. So we actually partnered with a colleague at UVic who then harvested some endothelium or epithelium for us, infected them with flu in the dish, and sent us some frozen cell pellets on 24-hour infection and 48-hour infection. And we get some really wonderful protein traces. So we're seeing things that are upregulated, downregulated. For me, this is as clear to me as math is. I mean, I'm a biologist for a reason. But there are some exciting hits in here. So we're seeing, vesicle organization, we're seeing viral life cycle. So some exciting things in here. We're still mining it. But we also quickly realized that this might not be the right approach, right? We might be able to see if there's a thrombin pathway in epithelium, but that's it. I mean, what we were just looking at with IVM was a little more complicated. So instead of just getting the lung epithelium, we really need to get into the same picture, the virus itself. We need the immune cells. We need the end product, whether that be cytokines or fibrinogen deposition, whatever. We need the whole picture to understand what's happening. So Tony encouraged us to do whole organ proteomes, and he's very proficient at this. And in the literature, nobody's ever looked at an entire mouse lung before with influenza infection. They've looked at cells washed out of it, or lungs that have been stripped of immune cells. So we took a very simple approach where we infected our animals or a PBS control, and we actually harvested the whole lungs, adherent leukocytes, things in the BELF, everything is one package, and submitted that for proteomics analysis. And this is still early results. We're really getting into the analysis of this now. So we're by 5 days, we're seeing this wonderful upregulation of proteins. And when we go to these heat maps, everything you would expect, complement and some coagulation factors. So it's exciting, but it's overwhelming. Like I don't know where you start when these are all pathways, and these pathways have 20 or 30 targets per pathway. So Stephanie in my lab started doing some string DV and tried to put together these interactive models, which make about as much sense as the heat map does to me. But it was cool, right? There's lots of dots and even more lines than dots. So it's pretty excited as a lab. For those that are old enough, this is Tinker Toy. So having flashbacks to my youth. But Steph was quite nice in that she knows that I'm slow and put on labels for me. And we can see some key pathways getting activated here, some key targets. So we've got complement activation, we've got pyroptosis, DNA replication, coagulation, oxidative burst, peptide trafficking, vesicles and stuff. So I asked Stephanie, well, where are we going to start, right? This is a wonderful, rich data set, but where are we going to start? So we want to go in and intervene and come up with a therapy. Where do we tackle this? So Stephanie went and on her own set up this really engaging set of criteria. So she said she only wanted to look at drugs that already exist. We are not a small molecule lab. We are not bright enough to figure out the next whatever. So we're going to cannibalize other people's drugs and use them for a disease that they've never been used for. She wanted, and this is largely because of our animal care committee, if there's animal dose data available, we can get amendments done pretty quickly. We don't want to spend 4 years of her PhD convincing the committee that the drug is safe. We needed a simple route of administration, so IV, sub-Q, something like that. We weren't ready to start gavaging our animals. For first run, we wanted to keep this simple. And obviously, that hasn't already tried and failed for influenza. So in this, we chose 3 clusters to start with. So we started with the pyroptosis, complement, and coagulation cascades. And we also identified there's this wonderful little hub in here, which is plasmin, and that really feeds into both coagulation and complement. So we set out, so this little hub in here. So I asked Steph, let's pick some therapies, right? Let's see what's out there. So she elected to go with a tranexamic acid for a plasmin inhibitor, which should take out the complement as well as coagulation. She looked and found a small peptide, PUR4, which inhibits fibronectin association with integrins, and fibronectin is in the center of this hub. And then she also found disulfiram, which is a pyroptosis or gasdermin D inhibitor, and this is used predominantly for alcohol use disorder, but is a direct inhibitor of gasdermin D. So these potential therapies she picked, if we think about how they're going to work, so our tranexamic acid is going to prevent the generation of plasmin, which ultimately will potentially limit complement activation, as well as the fibrotic clot breakdown. Disulfiram is going to inhibit caspase 1 cleavage of gasdermin D, and gasdermin D, once cleaved, forms pores, and there's some theory that we get sublytic pores and platelets, and that can drive aggregation. And gasdermin D is expressed by platelets. So we thought we'd try disulfiram, and then finally this cute little poly amino acid peptide that directly inhibits fibrinogen associating with an activated integrin. It also prevents actually fibronectin from aggregating on its own, so forming bundles, but critically prevents outside-in signaling through these integrins, which also could potentially drive inflammation. So we started out on this, and myself, I was extremely pumped about tranexamic acid. I mean, as somebody who has worked in the liver now for more than a decade, that is the #1 go-to for chronic liver disease, where you've got clotting disorders, risk of bleeding. It's a drug that all my cool colleagues use all the time, so I wanted to be part of their club. So we tried it first, and it failed miserably. It didn't matter how much we gave, when we gave it, how we gave it. There was no, I don't think I've ever seen negative data as close together as it has no effect on mice, or at least on flu. So my favorite horse never left the gate, and the lab was more than happy to rub my face in that when it happened. So the boss is not always right. Next up, we looked at a couple others. So here's our disulfiram in green and our fibronectin binding inhibitor in blue. So the green one is this bottom one, the blue is here. Our argatroban is our gold standard that all mice survive flu. These guys are almost as protective as our argatroban, at least statistically. And critically, our argatroban is given before we infect. These ones are administered now on day 3 of infection. So we can see they start losing weight and then they turn it around. So these are functioning as a therapy now. We're intervening after inflammation has started. And again, this is really new data that Steph has been working hard on. So I have very limited follow-up on this, but we do have one metric, and these are, for example, the MPO, so the amount of neutrophils in the lung. Disulfiram is once again just as protective as the argatroban. So getting in there and inhibiting the pyroptosis, or at least the gasdermin D, after inflammation has started, is turning out to be quite protective. And now Steph is working on confirming this and looking at Westerns, is gasdermin D cleaved or not, is caspase 1 active or not, and we're trying to map the pathway while still pursuing the PUR4 as well. So a summary for this part was that, this global protein or proteome approach allowed us to identify a number of different pathways. We still have another 3 that we haven't even started to probe, but there's some interesting opportunities there. Using the bioinformatics, we're able to assemble these 2,000 proteins into pathways. And when we did that, there was actually a number of hubs where single proteins regulated multiple players, and they became likely targets. So again, there may be other approaches, but this was our first step in it. And in doing that, we've been able to identify perhaps some therapies that can be repurposed and are shaping influenza at the moment. But again, this is the tip of the iceberg, right? We need to now perhaps get into single cell proteomics to understand who's expressing what. I mean, if we're worried about gasdermin D making sublytic holes in platelets and it's not in the platelet, we're once again chasing a red herring. But this has really, to me, reinforced that we're actually chasing thromboinflammation and not immunothrombosis. But it also raised another question, is that that's also subcellular entities. So we're always focused on what we see with the intravital microscope, so neutrophils and platelets. But we know that flu, for example, generates massive quantities of microvesicles. And I just want to show, again, this is a little bit of raw data and some of the troubleshooting we're undergoing. So we started looking at microvesicles produced in flu. And what we find is that by day 3 of influenza, we can see a larger representation from neutrophil-derived ones, endothelial-derived ones, really no contribution by monocytes. But when we started looking for what could be driving some of these processes, and we're particularly interested in other sites of distal inflammation. So influenza is associated with myocardial infarcts and stroke and other things. So we started looking for tissue factor positivity on these microvesicles. And we're quite surprised to see that this is very pronounced by day 3 of infection, that these microvesicles, who normally are not expressing tissue factor, are now strongly expressing tissue factor. And this is something that can circulate and now take tissue factor to a fatty plaque in the coronary artery or something. So we wanted to try and, again, understand these, and we're quite fortunate to have Tony Dufour. So we've partnered and we've tried to do some proteomes now on isolated microvesicles. And once again, we get a wonderful confusing heat map that I just trust Tony. But we did start to put some string DB together, and we're getting some interesting networks. I mean, when we're looking at the processes involved, right, there's a lot of humoral immunity, there's a lot of innate immunity. However, when we started diving in, there was a couple of things that were setting off red flags for us, and that is we're seeing an awful lot of acute phase proteins. And these are soluble proteins. These should not be in an MV. These are in the blood. And it turns out that when we look at our list of proteins, our number 1 protein is actually albumin. So we're starting to realize that the presence of these things means no matter how many times we're washing EVs, we can't get rid of the serum proteins. And it's becoming a bit of a problem because it's masking our proteomes. So we've taken a whole new approach, and this is, again, being able to admit where Baby Simple didn't work, and we've put a new student on this, and she has spent the last year, and we are now quite confident. So she's now started pulling these EVs out with magnetic beads where we can wash them in 200 and 300 milliliters of wash over and over and over again. And when we do that, we see about a 90% reduction in albumin as measured by Western blotting. So we're hoping, and those ones are now in the queue as I left Calgary for proteomes, that we can start to get more pure on what the actual MV is. So again, quite exciting results. The fact that we can actually get a proteome from an MV from a mouse on a day 1 and day 3 post-infection, but a little bit of caution that just because we get protein doesn't mean it's as clean as we want it to be. So we've really had to invest in getting those cleaner, and I'm hoping in the next week we're going to have our first proteomes from those EVs. So again, we're coming back to this chart here, right? That we really need to be careful in how we interpret these things, immunothrombosis versus thromboinflammation, and specifically how we target it. And if we take one step back and go to the previous models we had generated, so looking at how this works in the liver, where it is a macrophage and neutrophil activator process, a platelet stock, make a net, and we get clots, versus the lung, where it's tissue factor, thrombin, ideally, perhaps platelets, although we don't know who else is playing a role, and then we kick off the pathway. If we target thrombin in these two infections, we get very different responses. So if we target thrombin in the liver, we inhibit immunothrombosis, but we don't quell inflammation. If we target that exact same molecule and exact same pathogen even, so Staph aureus in the lung, we shut down the inflammatory process entirely. So again, understanding specifically the infection, the location, and how these two pathways overlap allow us to, for example, block coagulopathy versus blocking inflammation. And ultimately, this is where we need to be much more precise in dissecting apart immunothrombosis, which seems in our hands to be largely bad, and thromboinflammation, which, if controlled, is what we need to clear infections. So we're slowly trying to wrap our head around what we've been studying for 10 years is probably not actually what we've been studying. And we've actually been in that thromboinflammation camp without ever knowing it. And with that, I'm just going to obviously thank the people who have done all the heavy lifting. As I said, all the proteome work has been by Stephanie. Agustina and Laura contributed almost all of the lung intravital data I was showing before. Sydney is our microvesicle expert and magnetic bead guru, and then everybody else over the years that have really contributed to making this collaborative work quite exciting and engaging. And with that, happy to take any questions.
Thank you for the so exciting data. I have two questions.
Sure.
The first is in the lung, so you see the influenza virus. Do you think it's this influenza specific, or does other virus also do the same thing?
Yeah, so it's a great question. Is it influenza specific? So I didn't show data today. We've done SARS-CoV-2 in the humanized mice, and we're seeing very similar responses. The bigger difference is, although we see neutrophil recruitment, platelet aggregation, we see nets, we see thrombin, we see everything. Turns out when we target thrombin in that system, we actually don't stop the neutrophil. So for flu, we stopped all recruitment. In SARS, when we treat it, we stop the nets and we stop the thrombosis we see. But neutrophils still recruit to the lung of the SARS mice. Now, we're not sure if that's a pathogen difference, or if, we do know that these humanized mice for SARS are not great models. The receptors expressed in the wrong places. So we don't yet know. We know that the model holds up for bacterial pathogens, but the only other virus we've tested is SARS, and there's a bit of a split. But again, I don't know if that's the humanized mouse or the pathogen. We've not yet tried any other influenzas.
And the second question, sorry. The second question is, so do you see the Cooper cells in the liver?
Mm-hmm.
Do you also see this alluvial macrophage and neutrophil with platelet aggregate together in the lung?
Yeah, so the alveolar macrophages, they're in a different compartment. So they're in the airspace. They're not contacting the platelets or the neutrophils that we see. There are some neutrophils that get into the airspace, and they do contact that. That's work largely by Paul Coops. So we've stayed out of the airspace. Paul's been looking at the alveolar macrophages, but we don't see platelets out there, and we don't see thrombin generation directly, or at least we've not had any evidence of it. We don't know whether, again, the Mackman group has suggested that there is a PAR4 activation, but that may be on the basal side of the epithelium. We don't know exactly where that is, but we're not seeing platelets. We don't see the fibrinogen deposition in the air spaces, and we've done that both by intravital and by conventional histology looking for fibrin, and we don't see it in the air spaces. Yep.
There we go. That was great. That was super interesting. So much to think about. So I'm interested in the Kupffer cells and how they might, initiate events, because they make lots of factors that regulate coagulation. Like, even though you showed tissue factor was not essential, they make that. They make UPA. What else do they make that might help get things started in terms of recruiting neutrophils and getting the platelets to coact to bind to the neutrophils?
Yeah, so one thing that we tend to overlook or not think about is they're also fully decorated in integrins and other adhesion molecules that become active. So in data I'm not showing, we know that platelets actually patrol the surface of a Kupffer cell. So under basal conditions, circulating platelets will touch down for 1 second and then leave, touch down and leave, touch down and leave. If that Kupffer cell becomes activated, LPS or bacteria, those platelets, when they touch, they don't leave anymore. So there's an instant switch somewhere in an adhesion molecule that now keeps that platelet docked. We know that the platelets degranulate and they communicate with the Kupffer cell and a whole bunch of processes kick off from there. But that early immediate response, faster than we can make a cytokine or there does appear to be adhesion molecule conformation switches that allow these patrolling cells that normally release and say there's nothing wrong, they now stay. And that's particularly true in that bacterial model. So if we mutate the GP1B, the touch and go still occurs, but the aggregation doesn't. And if we get rid of the integrin on the Kupffer cell, the touch and go doesn't happen anymore. So it seems to be a VWF mediated process. So the Kupffer cells are clearing the blood of VWF. They have it on the surface. The platelet can dock on it. If that cell's activated, it binds to integrins and doesn't leave.
So this should be defective in all kinds of knockout mice that block inside-out integrin activation.
We've not looked, but I would imagine it would be, yeah, that those platelets binding, so they'll both be mechanotransduction pulling on it, they'll be outside in, but there will also be directional degranulation. And we believe that that's actually how the platelets might be, the candidate molecule is one of the defensins that actually activate the neutrophil. So why do platelets landing on the surface of a neutrophil trigger a net release? We think it's actually because when they degranulate in the circulation, everything diffuses away. When they're on the surface of the neutrophil, they degranulate right on the surface and we get a very different response.
Very cool, thanks.
Yeah.
I have another question about the integrins. I didn't note which integrin it was that the PIR4 was targeting. Does it have anything to do with immune cell migration into the lung? Like if you looked later, are you going to affect adaptive immune cell recruitment and viral clearance?
We've not. The PUR4 was applied 9 days ago. So we're still really early in the results. So it does. I mean, the literature that's actually using it is looking at blocking immune cell migration into the brain. So this is actually being tried in some degenerative brain conditions. And that's where we found the optimal dosing. So it is interfering with integrin binding on multiple leukocyte subsets with the fibronectin network, as well as potentially other ligands. So it is binding specifically to the fibronectin itself. And is occupying that docket, but it's also preventing fibronectin bundles from forming, which are then messing up other adhesion molecule sites when they're not forming the bundles. So it's both direct and indirect by changing the fibronectin network.
So in this context, we titrate active pathogens, see if this has a functional effect, because that seems useful, or is it just look like might provide protection?
Yeah. So you've been talking to the lab, haven't you? So we've titrated the influenza, and turns out our secret strategy of inhibiting thrombin that saves 100% of our mice with a lethal dose of influenza. So we're giving 250 PFUs. The mice are dead by day 6, or at least reach a humane endpoint. If we give them the argatroban, 100% of our animals survive. If we dial back that influenza dose to 50 PFU, which when the mice are not treated, they lose about 12-14% of body weight and bounce back and are happy. We give them argatroban, they're all dead by day 3. So this is where we were talking with the Mackman group. And the Mackman group has indicated that at least low pathogen challenge, you require a thrombin-mediated signal to increase expression of TLR3, TLR7, so the body knows the virus is there. We're giving a big enough dose that there are other PAMPs that alert the system immediately. At the low dose, if you're not having optimal TLR3, then the virus sort of gets a day or two to get a foothold and start replicating. So we're at the extreme end where we're not relying on a single DAMP or PAMP, that there's enough in there early on that toning down those receptors and the overall immunothrombosis is protective, or sorry, thromboinflammation is protective. If it's a really mild dose, you actually do need that thrombin signaling for sensitivity. So yeah, we've not figured that out, but it's my favorite part when I actually show this stuff to high schools, because we've cured flu, and if you go to your doctor, he's going to ask you, are you really sick or just sort of sick? Because if you're really sick, I can help you. If you're sort of sick, you're dead, right? And you find a lot of kids still go to school when they complain about, no, you're not. So we haven't quite figured it out exactly downstream, but we do think it's TLR3 dependent. We've not done it for the other pathogens.
Thanks
I know
for-
yours is going to be hard. I'm avoiding it.
Thanks for a beautiful talk. I didn't appreciate sort of this, immunothrombosis and thromboinflammation differences before, and kind of going back to the COVID and SARS-CoV-2 infection. So severe COVID is, associated with coagulopathy, thromboinflammation, all of these things happening much later.
Yeah.
That really causes severe disease. And so, but presumably there's, maybe based on your data, likely some of this immunothrombosis or thromboinflammation going on early,
right,
in infection. And so I guess, what do you think? Is there a crossover that happens at some point?
Oh,
or
absolutely.
Is there a switch that, and what do you think is happening in sort of, severe disease?
Oh yeah, I mean, you really can't have one without the other, right? You're putting active thrombin in the vessel so there will be fibrinogen, fibrin conversion, there will be microthrombi, right? Likewise, when you're clotting, you can't help. I mean, even under hemostatic conditions, you're activating the endothelium, you're activating neutrophils, you're activating. So there's always an inflammatory component to it. I think what's happening with some of these diseases is we lose the regulation of it. And I think what we've been concerned about in the lab is we've maybe been chasing the wrong thing, right? We've been focused now for the better part of 10 years of how do we bust that clot and reopen the liver to circulate. Don't worry about the neutrophils. It's the clot that's causing liver damage. And that might be the case in places such as the liver, but clearly in the lung, I mean, the clots are dispensable, right? If we administer high dose argatroban starting on day 2 all the way through, we do not see platelet aggregates. But the animals still die, and the lung it's massive fluid retention, and so there are 2 separate things happening there. And in some places, the clot itself may be the damaging one, but not always. And it really then shapes how we therapeutically intervene, right? If we stay focused on the clotting in SARS or in flu, we're missing the other side of it, right? At the same time, if we target just the inflammation, as pointed out with the low pathogen challenge, we could be doing more harm than good. Inflammation is a protective response if it's controlled, so we can't just plow it down to nothing. So we're really trying to find that nuance, and I do think that they coexist. And I just think we've actually been studying it for 10 years. We've just been using the wrong label on what my lab's been doing. No other answer.
Okay, I got 3 short answers for you. So first one is that bouncing of the platelets on the macrophages. That looks like catch bonds. Is that P-select and PSGL1 doing that?
It seems to be again von Willebrand factor. So
Okay.
it was
I
the
was
work
gonna
by
ask you
Connie
if you
Wong.
throw
So it was
that-
GP1B and von Willebrand factor were driving it, and then there was integrins on the macrophage that were required for the larger aggregate. So once they dock, it was switching over to the Kupffer cell doing it, and
Okay.
CD41 on the platelets.
I'd still be curious to see what happens if you throw an antagonist for P-select and on there.
Yeah, I mean, the other one is just slow the blood flow down a little bit and just see if we really mess them up, right? If we get rid of their shear or speed it up a little bit, which is easy to do in the mice, do we mess them up? Yeah.
And the second one was, I think you said that if you block the formation of these kind of nets in it, that they still clear the bacteria.
Yeah,
So there seemed complete, so then throw an aspirin in a person with sepsis is gonna be the trick to, and it's not gonna
Yeah.
have any influence? And
Yeah, I mean,
where do you see the bugs go then? Do they
Well,
do they clog up the kidneys or?
no. So when we give the net, when the animal makes nets, a lot of the bugs get caught in the net, right? When the net is gone, the Kupffer cells actually grab all the stupid bugs. So the nets are actually hiding the bacteria from the Kupffer cells. I mean, killing them themselves.
Yeah.
But we have to keep in mind that our mice are essentially the equivalent of what, an 18-year-old healthy, right? I don't know how my liver works after a night at the pub.
Yeah.
And in which case a net might save my life because my Kupffer cells are still drunk, right? I mean, we don't know. Like this is an optimal model where there's no other immune deficiency. In that case, the nets are dispensable.
Okay.
But they're obviously there for a reason. That there must be other, changes to the system in natural models
Yeah.
where the Kupffer cell can't keep up, or the red pulp macrophage can't keep up, right? So the nets are taking some of the load. Now, at the same time, nets are extremely protective in soft tissue infections, right? So if we give the same Staph aureus subcutaneous and you don't have nets, it is found in your kidneys the next day.
Okay.
If you make nets, we don't find it outside the soft tissue. So within those soft tissue infections, nets are highly protective. Within the vascular system, when you've got a working liver and Kupffer cells. Now, the other one that we didn't show data here is other pathogens, Salmonella, Kupffer cells can't catch. They don't see them.
Okay.
So those ones all end up piling up in your spleen and cause clots in your spleen. So in those ones, nets may be highly protective. The Kupffer cell doesn't have a ligand on the Salmonella, but if you can get inflammation, the nets will still stick to Salmonella, ligand independent, right? So there may be pathogens again. So Staph aureus we use, E. coli, they're great. The Kupffer cell deals with them fine, but there's probably pathogens the Kupffer cell doesn't deal with. So the net is your backup mechanism. Okay.
And then the third one is if those bugs are bounced around on the vessels, when you get rid of the nets, do you start seeing vascular leak? Because you start getting kind of the endothelium activated,
Yeah.
and making gaps.
Surprisingly not. Like again, the Kupffer cells are so good that what we see is, and I didn't put the data up, it's in the paper, that the shift in the number of bacteria per Kupffer cell actually go up when the nets disappear.
Okay.
So the nets seem to be grabbing them in the circulation, but then holding them, and the Kupffer cell can't reach the bug, right? So they're sort of trapped in the net. And when the net's missing, the Kupffer cell that clears everything in the 6 minutes is actually still doing its job and catching the circulate, at least with staph and E. coli, but other bugs may be different. Yep.
Thanks, Craig.
Thank you, Craig. Easily the prettiest talk we've had in the past couple of years.
The slides, right? Not.
No, you, man. I'm really interested in your perspective on the role of complement. And I am especially curious about how your networking proteomics analyses linked the role of plasmin and complement, since the literature is a bit controversial in that area. Says first plasmin activates complement, and then in seconds it inactivates
it,
some of the factors in complement. So we don't really know whether it's a pro or anti. So will you be looking specifically for activation fragments and timing it?
If I had my say, I would love to look for the activation fragments. Stephanie's committee is already mad. She's looking at 2. Focus on one, solve it. Don't. So I might have to get a new grad student to take on yet another pathway, but it is something we're quite interested in. We do know that, for example, activated C3, so C3b, also drives net formation. So C3b interaction with neutrophil is at least in the soft tissue, is the predominant driver of nets in soft tissue. It's a little redundant in circulation, as it seems the platelet can do that job. But we do know it can contribute. It can definitely contribute within tissue parenchyma. We've not looked deep into the lung parenchyma. There's not a lot there, but there's obviously the possibility of nets and damage in that area. But we've not ever ventured down that road. We did some platelet work when I was with Paul, and it just always felt messy. The mouse knockouts were not very reliable, at least for our Staph aureus models. But if there were some suggestions on how to start tackling that, I'd be very interested in taking a look. Right now, we focus largely on the pyroptosis and the possibility of sublytic pores on platelets is where Steph is running with it at the moment. But those other pathways are sitting there waiting for someone to pick them up and see what their contributions are as well.
And I'm also curious about where your flu is produced. What kind of cells?
So full chicken egg cell.
So they're not really what we would be breathing in a
no, and I mean it's a
infectivity
mouse
model.
adapted flu as well, right? It's the PR8, so it's been passaged through about 30 generations of mice.
So they're not covered with a human envelope, which
No, they're not covered with
which
the,
may
no.
contributes to infection and
Yeah.
even where the virus is going to be
Yeah, and
going.
for that matter, they're not a mouse envelope either going into a mouse, right? So yeah,
Right.
they're coming out of the chicken eggs.
Yeah.
And then purified from that. And we've never, it's a great point. We've never even considered that about passaging through a mouse line to coat them with mouse. It's a fantastic point that we've never, I hope you're not reviewing any of, it's a massive flaw now that I'm thinking about it. Yeah, we've never done that. We've worked with the Korean lab in VetMed on who does a lot of avian influenza. And that was our mentor in growing our PR8.
Not the same.
No,
Excellent.
I get it.
So beautiful. Thank you
Oh.
so much.
Thank you. Thank you, Kelly.